I'm so excited to be here. This is kind of the best part of all this work is actually to work with all you guys doing this work on your own farms, collaboratively together, really exciting projects. So welcome to the Plant Breeding Projects Virtual Showcase. We have a few people presenting, and then we will have some time for Q&As at the end. Um, I just wanted to, I'm calling in. My name's David Katzel. I'm the BC rep for the Bauta program. I'm calling in from a Kwantlen Stolo First Nations land that is unceded territory on Turtle Island. And I just like to remind ourselves that we're here, we're talking about seeds, and like land, seeds were unceded as well. Um, and something that I often think about when I think of the different ways of thinking about land from a colonial or indigenous perspective, there's also a different way often of thinking about seeds. And the one thing that am more and more aware of is colonially when we create, because we're talking about breeding projects and creating new varieties of crops and what I often think about is there seems to be a trend that colonially we name our new varieties after people. And the indigenous way of naming varieties is a lot more again, I'm not an expert, but from what I've seen, it's a lot more place-based or story-based. And it's not so much about the person breeding the variety, but the variety itself and where it's from. And so as we continue our sort of journeys in decolonization of land, I like to think also how we might move forward and decolonize seed. And so just to let everybody know, the order that we're going to hopefully present in, we're going to hear from Fiona Hamersley from Metchosin Seeds. She's going to speak for about 10 minutes about her Swiss chard breeding project. We're going to hear from Dan Brisebois for about 20 minutes about some lettuces and radishes. We've got Chris Sanford from Yonder Hill Farms, who's going to talk about sweet peppers and tomatoes. And then Craig Boychuk of No Coast Seeds going to talk about some zucchini. So without further ado, I'd love to, if you're able to share your screen, Fiona, and jump in.
Yeah, thank you for inviting me. Nice to see some familiar and some new faces. And yeah, I was going to talk today about chard breeding. And um, first of all, like David, just really important to acknowledge the land that we are farming on and the fact that that land, certainly on Vancouver Island where I am, I benefit from thousands of years of cultivation that was practiced in the Garry Oak Meadows, lovely deep soil, etc. And we take the stewardship and the reciprocity of that very seriously. And we often have Indigenous and non-Indigenous students with us here at the farm, and that educational part of relearning some of these skill sets, whether it's cultivating camas or cultivating more recent introduced crops, is really important. And it's part of that journey of reconciliation, and we're excited to be able to do that work. It's a privilege. So we have Metchosin Farm, and why plant breeding and seed saving I thought was a good place to start. Why are people like us involved in this crazy journey that we're on? David, you and I talk often about the joys and challenges of small farms, and I'm sure there's some days where we think, "Oh goodness, why are we doing this?" And so I think it's good to remind ourselves of why. Certainly on a global and North American, Canadian scale, we've lost so much diversity in our food seed systems. So it's estimated we've lost about 94% of our North American food seed genetics in the last 100 years. The United Nations, FAO, says it's about 75% in the same time period. So I think that it's not just trying to hold on to and preserve these heritage varieties, but for us at Metchosin Farm, it's also about tinkering and creating new varieties. So, those kind of heirlooms of tomorrow. And you don't have food security unless you have seed security. And we know from the research that Seed Change put out, I think about 2 years ago, that we import an estimated 97% of the vegetable seed grown in Canada every year. Houston, we have a problem. So that's one of the reasons I think that um, the group of us are involved in this. It's super important work. And I think it's also about relearning and then sharing this skill set of not only saving seeds, but preserving seeds and learning to work with them and creating these new varieties. And that also, I think, is really important. So why chard? It's not the most obvious plant to be playing with. It's a very interesting set of genetics to work with. I love the diversity within chard. It's an outbreeder, so it's a bit of a challenge. You need that larger population. I like to work with what I call a working population. It is a challenge also that it's a biennial. It tends to cross with other things like beets. And also, I have a family history of my mom is British, was born in England. I grew up partly in the UK. My grandparents had chard in their garden when I was a kid that was sweet and tender and succulent. None of this old beet green nastiness in the back of your palate of oxalic acid that we just take for granted as part of the chard flavor profile, if you will. So in North America, we have rainbow or bright lights are the dominant sort of mixes. Those were originally bred out of a German Lutz winterkeeper beet, so that hence the higher level of oxalic acid, hence the wonderful colors though. So on the left, you'll see a silver beet chard, which is what my grandparents had at their farm in England, and that was bred originally out of a sugar beet. So that has higher sugar levels. It's more tender, it's more sweet, it's got the wider stems, but it's missing some of the key things that we love in chard, which are the bright colors, which are also very good for you because they have the pigments in them. So what I've been doing is crossing these two together for 10 or 12 years now, I guess. So it's a it's a bit of a project. I've been selecting the ones that self-seed in my field garden. I will occasionally take the seeds from an interesting variant that comes up and save those specifically and seed them out in a bed. But for the most part, I just let the chard run wild all over the garden. And as it grows up after its first year where it's self-seeded, if it meets the criteria that I'm looking for, then in year 2, I dig up those seedlings and I transplant them together into a 50-foot bed. So I'm looking for flavor, for sweetness, low oxalic acid, so that bitterness in the back of your mouth, and wide, large, juicy, tender, sweet stems, because to me, that's what chard means. I have size 8 feet. So that's a pretty big piece of chard. You'll also note some of the color variants in it. It's got a dual tone stem, which is a recessive trait. So that's harder to stabilize, but it's definitely in the working mix. I'm also looking for disease resistance, winter tolerance, and beauty. So on the left there, you'll see a seedling that has obviously a higher amount of the silver beet chard in it. And on the right, you'll see one that's a mix, has the wide stem on it, that's doing very well in sub-zero conditions on the coast here. I'm also looking at which of the seedlings that come up are quite resistant to Cercospora spot, and I've noticed a good trend in that over the years. Looking for dual tone stems, again, that's a recessive trait. It's harder to stabilize, so the best that I can hope for at this point is just keeping it really active within the population that I have. Those are photos of stems where I've cut the stem and then flipped over the bottom part of it, so those are all from the same stem. I'm looking for really good quality seed, obviously. I'm looking to maintain a diversity of colors. Orange or yellow, for example, are not dominant. And so it's important when I plant those out into the bed, I put those in chunks within the bed. They still cross-pollinate with the other ones, but they're closer to each other. And I'm also looking to select and encourage unusual variants. So my approach to this, I'm fitting this project in amidst a pretty full farming life and family and other commitments. And so I have a pretty casual, fun approach to it. This is this is one of my fun things that I'm doing at the farm. So I don't take it too seriously. And some years I don't grow as much as others. And some of the other varieties that I've been working on, like the strawberry cherry tomato, was a more specific cross using paintbrushes and bagging it. The chard, I just let it run free. And I really enjoy what I think of as the artistry and the alchemy of this project. You never know what's going to germinate and pop up and what it's going to look like. And it's pretty exciting. And again, at this point, I'm using more of a working mix with this project. So I'm not looking to stabilize anything really specifically. That's Ben when he was younger, and he's got silver beet chard on one side and a perpetual spinach, which has also been crossed in on the other. And yeah, I don't know if anyone has questions.
Yeah, I think what we'll do is we'll go through, feel free to throw your questions in the chat, and if it's okay, we'll go through all the presentations and then can do questions at the end, unless anybody has burning questions they need to ask right now. If you
Sure.
could plop them in the chat, that would be great.
Okay, and then David, if you'll bear with me just 1 minute, I'm just going to quickly stop the share and bring up one single photo that is an interesting variant that I forgot to include.
Okay.
So a couple of years ago, there was a chard that popped up and we lost it. And then a few years later, one of its offspring popped up as well. And it has now turned up a few times in the garden. And I'm just really excited about the potential with it because it's such an unusual mix of colors and ruffled stem. So you can see that there. Do you see the stem on this one? It's just the most gorgeous sort of deep ruffled, and it's probably 1.5 inches wide. And so I'm really excited to be potentially turning that one into a single varietal. And it's striped like a pink and white peppermint on the other side. So stay tuned.
Great, thanks so much. All right, I think we're going to hear from Dan next. So,
Okay. So I'm Dan Brisebois from Tournesol Cooperative Farm. I'm going to start with some slides, tell you a bit about who I am, and then I'll jump into some breeding projects. So I'm part of Tournesol Cooperative Farm. We started in the fall of 2004 with 5 folks who met studying agriculture at McGill University. At this point, we now have 10 co-op members and 10 non-members, so about 20 people on the farm, and hopefully more of the non-members become members over time. We're situated west of Montreal in Les Cèdres, about 45 minutes west of Montreal. These are our fields. And we're vegetable growers. And we grow about 7 acres of vegetables in any given year. And we have 500 weekly CSA baskets that we fill with the vegetables. And we're also seed growers. And we grow about 1 acre of seed in any given year. And we sell it through our online store, the Seed Rack program. These are our packs. This is our farm website, ferme.qc.ca, fully bilingual. And I'm also the co-host of the Seed Growers podcast. These are some of the fine folks who were guests in the first season. And now I'm going to talk a bit about some lettuce breeding that I've been doing in recent years. And this is a more recent project. And then I'm going to talk about radish afterwards, which is a project that has been around for a lot longer. So we grow a lot of lettuce for seed, either for our own farm use or for sale through our seed company. And we grow lettuce with next to no isolation distance. So what you see here, there's, I don't know, 5 or 6 varieties that are almost all side by side. And we don't see much crossing with them. And, they grow up and we grow them to get seed. And every now and then we'll see one that's going to come up that's clearly different than the others. And that was a crossing from a previous generation. So there's one that we have that is a crossing from some deer tongue and a romaine lettuce that we found in 2001. And I can't find a picture of what it actually looked like, but it was from this generation. And so what we did is we had the seed the mysterious seed popped up. We flagged it in the field or in the greenhouse, and just made sure to keep the seed separate from it when it was time for the seed harvest. I put it in an envelope with a label, and then the next year I grew out that seed. And this is what it looked like. So here you can see there's a bunch of green lettuce, essentially, that have different leaf shapes popping up there. And I grew about, I don't know, but I grew about 30 or 40 plants. And we would walk through regularly and say which ones we liked the best, and we flagged our favorites. But ultimately, what I let happen is I let them start to die back, whichever ones weren't doing well. So we grew them out in the field the second year, and so we live in a wet climate, and the ones that weren't disease resistant would die back, and the ones that were disease resistant would survive. You can see the ones that are flagged are some of the ones that we liked the way they looked, and then the ones that aren't flagged are ones that nature liked. And those are all things that survived. And we kept, at the end of the year, I had, I guess, 7 plants that had survived through the season. And whether they were flagged or not in that year, I just kept the seed from them in 7 individual packs. And then in 2023, which is this current summer, I grew out. So this is a field of, this is again in our, this is our so the year 2022 was in the field. And this year we grew them out in the greenhouse, and I grew 10 or 15 bed feet of each one. So something like maybe 30 plants from each one to see. And they're kind of divided by different, those pink flags. And then some of them are also, there's also some variety trials in there too. And so here's kind of, you can see 2 selections side by side. So these would have come, what was on the left side came from the same seed pack, and on the right side, a different seed pack. But the year before that, they were from the same plant. So they're, at this point, I guess they're first cousins. And in some cases, some of these groups, these populations, so some of them looked really, really similar one to the other. We grew them on 2 rows between, or 2 rows per bed, and 9-inch between plants, which is why they're really tight together. And I like to do that so I have a little bit more space to kind of see between the rows, and then I can rogue out the ones I don't like between the plants to give them more space. But this here is fairly uniform. And then there was others that had a lot of diversity in the populations. And at this point, we went through and started to choose the ones that we liked the most. And so there was one that I really liked that was fairly uniform. And the way we chose what we liked was based on the looks, and based on the taste, and based on how, whether it was bolting or not. And so this was the favorite one. We do not have a name for it yet because this is just in the F3 generation, but it's a nice lettuce that we're going to try growing out in our market garden this summer. And then we'll also grow out the F4 seed to see what it's like. And then we had some that, like, so this is a row that had one that was very different from everything else that I really, really liked. And my wife thinks I like it just because it looks like tatsoi, but whatever the reason, that was the one my favorite. And this one, unfortunately, didn't make the cut. It succumbed to disease, and I'm very disappointed that we didn't get any seed out of it. So I'm going to go back to that group and try to see if I can find another one out of it. And so what's my next picture going on? Yeah. So at this point where we're at with this is we're just growing out every year what we like and getting rid of what we don't like. And I'm assuming within 3 or 4 more years, we'll have something that's fairly stable and that we'll be happy to release, maybe even before that. And we'll also start growing in our farmers market, and in our market garden for CSA. And that'll be another round. So if it's successful there and we like it, that'll be even more reason for us to keep growing it. And so in the meanwhile, I'm always looking for others that have escaped that cross lips. This is one that I found in a bed of I think of sucrine lettuce. I found one just that had a kind of has an oak leaf type with spots on it. So I know this is an F1, probably between a red salad bowl and the sucrine kind of like just like butterhead romaine lettuce. And so this is something that next year I'll grow out in the field to see what survives in the field, and then kind of continue the process. So that's just a little bit of the lettuce that the lettuce work I'm doing right now. So I've selected lettuces in the past And something I found overwhelming is how much different diversity you can get. So I'm early trying to eliminate some of it through having it just succumb to disease to kind of get to what's good quicker. So I'm going to wrap up the lettuce and start talking about winter radishes. Should I leave space for any questions, David, or should I just move on?
I think you move on, but people can feel free to add questions to the chat, and we'll keep track of who the questions are for at the end.
Perfect. So this last project, so the lettuce, in that case, I was working with something that I found randomly in the field and decided to keep. And a lot of breeding projects on our farm start that way, but then others start with a lot of intention. So this is something that we started, I started maybe in 2007, I think, the fall of 2007, or maybe spring of 2007, where I planted some watermelon radish, which is on the left with the green outside and a pink inside. Or I should say, we harvested some watermelon radish, which is what's the green outside and the green outside and pink inside, and we harvested some black radish, so black on the outside, white on the inside. We harvested them in the fall, overwintered them in our cold room like we would for any storage route, and in the spring I grew 5 or 6 of each radish side by side, and just like 2 little plots side by side. I let them go to flower, and I let them cross-pollinate, and then I harvested the seeds from them. And there was only a short window while they were both in flower. One, I believe it was the watermelon radish, flowered a lot quicker than the black radish. So it was only a short window where they were able to cross-pollinate each other. And I could definitely see that when I grew the seed out, because when I grew the seed out, most of the seed that I'd saved from the black radishes were mostly black radish, and the ones that I'd kept from the watermelon radish were mostly watermelon radish. But then amidst there were a dozen or so of these fantastic, maybe a little bit ugly, radishes. So this is the F1 hybrid between the two. And these were purple on the outside and purple all the way through. And some of them had a little bit of that black rind, kind of like a black radish. And I kept that seed. So I grew them. I grew a row of each, and then I kept all the ones that had been crossed up that I knew were F1, and then I overwintered those together. And the next year, I just planted that group and let them cross together. And then I kept the seed, and in the fall, I grew that out. And then this is what I got. So I got this diversity of different radishes, some that were completely white on the outside for the inside, and then I got some that were pink on the outside, on pink in the inside, and then some were black on the outside, and there was some pink or white on the inside. And this is still the same batch. And there was also a lot of purple that started to come out of the population. So this is the F2 generation. There was a ton of diversity, and it was really, really exciting. And I kept the ones that had black on the outside. I could see color through them. And the next year, I grew them out. And so this is not necessarily the next year, but basically I grew them out every year, keeping what I liked. And over time, it became more and more stable. And so then these are some of the phenotypes that we're seeing with kind of purple on the inside, pink on the inside. And what's interesting with this is if you go back to the watermelon radish, there was no purple in any of this. There's that pink for sure, though it's a little bit of different pink, but I think that pink has some, that's where the radish, the purple radishes are coming from. And so that's something that kind of got unleashed by this cross that wasn't expected beforehand. So what it kind of looks like in the fall when we harvest is we have our rows of winter radish, and we grow them 2 rows per bed, again, so we can see them a little bit easier and let them size up better. And then I pull everything and leave it down on the ground. And then I walk down the bed and I select everything that I want to keep for seed. And I leave the tops. And this is maybe a little bit too much tops. Maybe I like to have like maybe more than an inch or 2, but I don't want to cut them flush with the top of the root. Excuse me, because I want the growing point to survive. And so at this point, I'm just basing myself on what the outside looks like. And if I can see a little bit of red through the black, then I'm going to keep it. And then the ones that don't look like what I want, I cut those leaves off, and those will go to the CSA and get eaten. So there's a lot of off types that come out and get moved to the CSA. So here are different selections I've been making. because with this, there's been so much diversity. I've begun to work with more than just that one black outside, red inside. And so the overwinter in the cold room, and then in the spring, we take them out and then we cut these little Muppet mouths on them to see what the inside is. So we cut them deep enough so we can see the inside of it, and then we'll separate into, well, anything that really is completely white on the inside and we were fooled, that just maybe goes in the compost. And then the ones that were the darkest pink, we kind of have it as our A grade. We have some that are in between as the B grade, and then some that are a little bit less pink on the inside as C grade. And we'll grow them all out for seed in the field, but we'll keep our stock seed only from those that have the darkest. And then but we have the larger amount so that we're keeping some genetic diversity in there, just so we don't bottleneck it. And so that these are some of the ones that we would have selected to go out. And then we plant them in these little 3-inch pots because our spring can be fairly cold, and we want to make sure they get going and break dormancy before they're going to be planted out. So we plant them in these little pots about a week to 2 weeks before they're going to go in the field, and then we get them out of the field as soon as we can. We don't like to leave them in the pots too long because they get really, really long and stemmy, leggy. And we've known from the past that at that stage, if they don't get enough moisture, they are much more susceptible to dying in the field. And we've lost a couple generations that way. So get them out in the field quickly. So at this point, so on the far left here, you can see the traditional black radish. In the middle is what we call our black crimson radish, with black on the outside, red on the inside. And on the far right is our black amethyst radish, which is black on the outside, purple on the inside. Again, here's the black amethyst. And then so I'm also keeping all kinds of other things coming out of it. These are our skunk radishes, black on top, white on the bottom. Inside can be white, but some of it's pink. And I don't know if there's really a market for this, but I love them. And then we have all kinds of diversity that we've been working with. One of the things we've started to cross in is some longer genes from like some green Louisville radish. This is our kaleidoscope mix. So we've actually been selling the radishes as a crossed up mix too, instead of just what the end product is going to be. So that is those are our seed radishes. This is just some final slides. I have some seed resources at seedgrowers.farm where you can see different spreadsheets and workshops I've given related to seed. And you can stay in touch follow my blog at danbrisebois.com, or on Instagram, I'm at danbrisebois.farm. So thank you.
That's awesome. Thanks. Thanks so much. Those are so exciting. Okay, I think next up, and again, please feel free to throw your questions in the chat. We will we'll have them at the end, and we'll be able to ask Dan to elaborate on some of the answers. I think next we have Chris from Yonder Hill Farm.
Hello, can you hear me?
We can hear you, yeah.
Oh, good. Yeah, so I figured I'll just give a little intro about our farm. We're in Nova Scotia, so the South Shore, down the coast from Halifax on the Atlantic side, on a lovely Drumlin Hill. It's an old heritage farm. And my partner and I got into seed saving, thinking about sustainable farming practices and lifestyle practices, and just got more and more into it, I guess. So we bought the land in 2011 and started producing seeds right away. We had established a relationship with a small scale seed company that sells exclusively maritime grown seed called Annapolis Seed. And we've been growing for them before we bought our farm, and just got more and more into it. So now we've started our own website. I guess this is 5 years in with our own website. We have about 200 varieties or so that we're working with. We do it all ourselves. We don't do any buying and reselling, so it's a lot of work and not the most profitable, but a lot of fun. So I thought I'd share. Steph Hughes, our Bauta family initiative rep, asked me to share some of our interesting breeding. Yeah, I'll say that, sort of starting out, I didn't really consider myself a breeder. And And then once you kind of get into it, you realize that you know, inevitably are selecting and things are adapting to your climate. And kind of the first time I realized, oh, I am breeding new varieties was, you know, working with actually a squash, and just through selecting year after year for early fruit, I was really advancing the ripening. What was a long season squash became a short season squash, and I realized that, yeah, that was my sort of beginning to realize that we're into plant breeding. So fast forward to this year, and we're releasing, it's more of a gene pool than a full-on stable variety, but we're pretty excited about peppers and also tomatoes. We do all of them outdoors, and we have not a lot of pollinator pressure. I've talked to other seed growers who experience a lot more crossing of their peppers and tomatoes in the field, but we found that not to really be the case. It's a lot more like what Dan was talking about with the lettuce, where you find the occasional cross, and it's kind of an exciting thing. I do separate with sweet peppers. Want to be absolutely certain that I'm not crossing them with hots, so I do physically distance those two types of peppers away from each other. And I have a little bit of physical distance in my planting. Between sweet pepper varieties, I'll typically have about 6 to 10 feet between varieties. And I have fairly low instances of crossing. And I believe it was in 2019 that I first noticed this cross, which the mother plant was a variety we've been working with called Napoleon bell pepper, which is a really nice bell pepper, probably my favorite of the types of classic bell, where it's nice and blocky and really tasty and crunchy and everything, but it's earlier than California Wonder. So we've been working with that one for a number of years, and this one plant popped up with this very different looking fruit. And so I knew it was a cross and sort of flagged it out of interest. And I pretty well knew that its father was this other sweet pepper variety that we work with called Clary Baby Cheese, because of the coloration of it. It's really the only sweet pepper that we have that is that color, sort of like when it's first coming on, it's not a green color, it's not a dark green. It is a very kind of whitish green, and then it ripens to red. So I was kind of excited about the cross and saved the seed. And I find with the peppers and tomatoes, and maybe someone with more, I guess, scholarly experience with plant genetics, it seems to be that sometimes you just hit it well, where the genes sort of are more stable right away, and other times they're all over the place. And I'll kind of get into what we've seen with tomatoes in the next few slides. But with this one, it was amazing, the F2 generation. So this first year, of course, is the F1, and then I grew out the F2. It was over 75% stable. I was pretty impressed. So something had lined up with keeping that kind of color and sweetness. It's a really nice sweet pepper. I brought a bunch of those peppers to market because I just had a lot of extra. And there was a chef who thought it was a type of pepper called gypsy pepper, which is I did some research into it, and that's an F1 hybrid variety, but apparently this one resembles it a lot. And let's see if I can, yeah, so this is from 2023, the F3 generation. And yeah, we had a really rough growing season, so pardon all the weeds in the picture on the left. Just by happenstance, this variety of pepper I planted just below this bed was a total swamp, and I lost a lot of crops. all the rest of the sweet peppers, and also my grow out of the pink peach tomato, which was supposed to be right next to this pepper. Completely succumbed to our wettest growing season that we've had ever, that we've been at the farm, and I guess for quite a long time just in general. So yeah, so I wanted to take a lot of pictures this year, and you can kind of see there are 2 beds of this pepper planting, and they're not doing the best. It was a pretty rough year for everything, but I did get a nice amount of fruit set from each one. You know, I was able to kind of observe the fruiting characteristics and found that there was even fewer off types, so it was really going in the right direction. Over on the right here, I've got the picture of what the off types were looking like. They had the dark green color, and there was less than 10% of the plants were exhibiting the off type. So because we sell our own seeds, and you know, we're not that big, I went ahead and saved a lot of the seed, and I'm offering it as a new variety this year. It's not 100% stable, but I'm pretty comfortable with 5% off type kind of as a general rule. So yeah, it's kind of exciting to us. It's our first sweet pepper, and they're really beautiful and pretty tasty. So we've named it Moon Cheese, just out of a kind of fun play on the cheese pepper as the parent. Yeah, moving on to our tomato experiments. We originally got this interesting gene pool from Fedco Seeds quite a while ago, in 2013 or 2014, called Pink Princess, and it's the picture on the left there. And they were releasing it as a gene pool, and I don't know if it still is, they still are carrying it as such. But it's a pretty neat cherry tomato that's super sweet, really nice color. And we fell in love with it and have been selecting it ever since. And some interesting things have come out of that gene pool. Clearly the pink color is genetically tied to the sweet flavor. So we select for what you're seeing in the photo there for the Pink Princess. But then the second year that we saved, or I guess the first year we grew the Pink Princess in our field, it seemed to have a lot more capability of crossing, I guess, with the flower structure and stuff. And so, yeah, we noticed that it had crossed, and again, we could really tell who the father was just based on the offspring. And it was clearly a variety that we have called Garden Peach, which is really fuzzy like a peach. And so what we ended up with was fuzzy like a peach, but pink and sweet like Pink Princess. And again, it stabilized quite nicely, even in the F2 and the F3. So we've been, selling that as a variety since 2017, and really love it a lot as a tomato variety. But we found that there have been some other Pink Princess kind of variants that we've tried saving, and they just haven't had that stability, and they just have been all over the place genetically. So I kind of wanted to just share from this year a lot of my experiment variants that I've kind of found over the years. Some of my seed was getting quite old, so this year I decided to just grow out a lot of the crosses to see if they're worth anything, any exploration. And I mostly discovered a lot of, lack of stability genetically, and these were basically all F2 generations. So I had saved an interesting cross, back in 2016, and then just kept the seed, and the seed's getting old, so grow it out, and then just try to label things well. And I don't know, there might be a few that of these paste varieties that I'm interested potentially in growing out, but we're just into experimenting. So that's sort of what things were looking like for our tomatoes. And it's just, we find it's, you know, when you're just taking a chance cross, it's hard to know, even in a self-pollinating vegetable, it's hard to know that it's going to end up stable or not. It might take quite a number of generations and not be worth it, or you might find that it is. So yeah, that's basically what I had to share.
Great, thanks a lot. And again, yeah, feel free to add questions in the chat if you have any for Dan or Chris, and we'll get to them at the end, and we'll move along. Yeah, we'll move along now. So if you want to, if Craig, if you're able to share your screen.
Alrighty. Cool. Yeah, so my name is Craig Boychuk, and I'm running a small seed company called No Coast Seeds, growing open pollinated seeds for short season gardens. Very interested in plant breeding, and I'm making available a bunch of like really early work in progress gene pool mixes, breeding groups and stuff. Just really excited to kind of get stuff out there, release and reiterate. I've enjoyed working with a lot of that kind of material that other people make available. So yeah, it's been fun to kind of do the same thing. Concentrating more in general on newer open pollinated cultivars, more so than like older heirlooms, although I do have a few heirlooms as well. But yeah, pretty interested in exploring new stuff. So, and I'm located near Calder, Saskatchewan in Treaty 4 territory, just kind of near the border between Saskatchewan and Manitoba. So I'm going to talk to you today about double happiness bicolor zucchini. So here's just a few pictures of what it can look like and hopefully will look like. This project came out of a mix called Zucchini Surprise, which is an interbreeding mix of 2 yellow and 3 green zucchini varieties. And I had 4 goals for this population. One was just to make a fun mix that creates a nicely varied zucchini crop. I just really like mixes, and yeah, it's kind of cool to have a bunch of different zucchini to choose from. I was also interested in searching for kind of unique yellow and green color combos, sort of in the realm of a variety called Zephyr. It's an F1, I think it's Johnny's offers it. But I hadn't really seen any open pollinated bicolored zucchini, so it seemed like maybe a cool opportunity to fill a niche where it was just kind of no one had really done that yet. Although it's possible it exists, I just haven't found it. And I also wanted to start working with controlled hand pollinations, and squash is just a an easy way to do that. Thing. Really big flowers, and yeah, it's fun and easy to kind of delve into that realm. I also wanted to begin researching zucchini color genetics, and so this kind of having a project to base that around would help me to kind of focus on what I wanted to learn about. So this is what Zucchini Surprise looks like. It's just kind of a big old mix. There's some Costata Romanesco was one of the green zucchinis, and the other ones are just kind of more solid colors. But it's yeah, it's making for some interesting sort of stripy combinations and like some nicely ribbed plants. I don't have too many bicolored plants in these photos here, but there's a few bicolors that show up as well. So in 2019, I did an initial planting of those 5 varieties and just let them cross up naturally. 2020, the F1, I got 2 bicolor plants, but only got seed from 1. And I started doing some casual hand pollinations. By casual, I mean I wasn't like taping the blossoms or anything. I was just kind of going around and encouraging plants to cross with each other. But yeah, so in seeing that bicolor plant, I was like, all right, I'm on to something. 2021, there were more bicolor plants, which was exciting. About 10% of the population was bicolor. And I did more casual pollinations, focusing more on combining green and yellow plants instead of just kind of super random. But again, wasn't really doing any taping or anything. And in 2022, I decided to branch off to the Double Happiness project and like really concentrate on the bicolor zucchini. So in 2022, which was probably an F2 generation, I planted out 3 lines, which you see below there, 3 maternal lines. Only one of them really did anything. That's from the fruit on the left. And I got a 1-to-1 ratio of bicolor to solid colored plants, which is pretty good. And the other 2 just didn't really do what I wanted. So kind of given the lack of bicolor plants in 2020, I'm thinking that it was most likely that line arose from a cross in that year between 2 yellow and green zucchinis, which would make 2022's crop the F2 generation. It is possible there was maybe a little more like some sort of a 3-way cross that happened over the first 2 years, but probably not, is my guess. So I'm just kind of going with that assumption. In some ways, I wish that I had planted out more than 3 lines, but the project would have been way bigger than I could handle. So it's good to put limits on stuff, but it's always like, I always get kind of sad about all the stuff that I might have missed. So yeah. But yeah, line 1 ended up producing 5 different bicolor phenotypes in total, 2 of which I was very interested in. And I did a bunch of controlled hand pollinations, both selfed and crossed, with preference to my 2 favorite phenotypes. But of course, I was just working with whatever flowers happened to be available, because it's not always, you don't always get what you want. So this is what my messy zucchini patch looked like in 2022. I put the plants a little too close together. They were fine health-wise, but it made it a little difficult to work with them. But yeah, so basically just flagging the ones that I want. And this is what it looked like a little later in the season, getting a little wind blown. But yeah, still doing pretty good. I basically am just taping the flowers with masking tape and tagging the plants with a little piece of twine, and then making a note whether it's a self or whether it's a cross with another piece of tape on the twine. That seems to work pretty good. I also limit the number of fruits per plant, which I am finding results in a much better seed set and better chance for my crosses to actually reach full maturity. So that's why you'll only see maybe 3 or 4 fruits per plant here. This is what I harvested in 2022. All the zucchinis on the left are the solid colors, and maybe some discards, and the one in the front, the ones in the front are the selfed and cross lines. So these are the ones of interest from 2022. And I'm going mostly for the like really big kind of half and half yellow and green colors. And these were some of the self lines from 2022. And they typically look pretty similar when they're small to when they're big. So I found evaluating them as adults to be just fine. So in 2023, I chose 9 maternal lines from those plants that I just showed you, 5 of which were selfed and 4 of which were crossed. I didn't note the individuals for the crosses, but I would be crossing up like, 2 bicolor plants with similar characteristics. Ended up with about 6 plants per line, which was, again, an intentional limitation, but kind of frustrating. The average ratio for the whole plot was about 1-to-3 bicolor to solid, so it went down a fair bit from 1-to-1 the previous year, but that might have been because of my sort of smaller population sizes per line. There's a fairly wide spread of ratios within both the self and the cross categories, so I feel like if I had a bigger population, I would have had more meaningful numbers and just a little bit better info to go off there. So again, I hand pollinated all my favorite bicolored plants and ended up with 16 maternal lines. half of which are selfed and half of which were crossed with a similar phenotype. And this year I made notes of both parents for each cross, like what they looked like and which plants they came from, just to kind of be able to track what's going on a little bit better. And I tried to cross plants that were of the same phenotype. So I'm now just starting to get into the genetics behind it, which is kind of a backwards way to do it. But originally it started out as just kind of a more of a random experiment. There's a lot of unknowns in the realm of zucchini color genetics. It's not just as easy as like a green and a yellow gene. I believe there's a bicolor gene, and there's the way that all of these interact, as well as some mutations can kind of be involved, and environmental factors. So it's kind of complicated, having a tough time wrapping my head around it. But the general impression I get is that I would be best served by aiming for homozygosity. So I feel like I might get better results working with selfed lines or with like really carefully curated crosses with phenotypes that are super similar, which would, going on the assumption that they have similar kind of gene combinations. So for this year, the goal is to, of course, increase the number of bicolor plants, which is a hope as well as a goal. I'm going to do equal numbers of selfed and cross lines, aiming for more plants per line, maybe 20, maybe more, depending on how much room, just to try to get a better idea of how I'm doing with ratio of bicolor to solid color plants. I want to, compare those ratios between the self plants for the past 2 years, just to see if it's improving, which I would expect it to. And for the cross lines, I'm curious to see if there's any predictable color variations based on the parents, or if it's going to just be a little more random. Got to keep researching zucchini color genetics, of course, and with the hope of getting a little more dialed into what I'm basically trying to make better selections and having a better understanding of what I'm actually doing. And adjust my expectations based on that new info. It's possible that I may not get a fully bicolor population. There may be like the odd solid colored plant. There may be, the first few fruits are not bicolored on any given plant. So yeah, we'll just kind of see what happens. And better record keeping, which I have been slowly doing as this progresses and as I see that I want more info and don't have it. So moral of the story, keep lots of notes. You're going to be happy about it. That's about it for that. You can get me at seedsecurity.ca. Free feel to shoot me an email or what have you. So I'm just hoping to very excited to see where this lands next year, and I think it'll be kind of a nice make or break sort of year where I'll either find that my methods are working great, or I'll find that it's still quite random and I need to be more specific in some way. So time will tell.
Thank you so much. I love the experimentation, and I love learning from the plants before diving into the academic work. Sometimes that's the best way to do it. There's information that the plants can tell us that you might not find on paper. And some of us learn very experientially, and some by books. And there's lots of ways to do this. Just keep in mind, plants are your teachers as much as people, for sure. So we're happy to take some questions. We have a few that came from the chat, and there were a few that came from the registration page. I have a whole lot that have popped into my head personally. There was a question. For some of the questions I think that were asked for Fiona are answered in the chat, but if you're interested in elaborating a bit, there was a question about how do you test for low oxalic acid?
You taste it. I mean, I'm sure you could find a scientific test out there online where you could puree it up and use some sort of a litmus test. But yeah, I just like to pick the stem and taste it. And it's about the oxalic acid. It's also about the texture, especially after like this recent cold snap that we had. Some of the stems still looked really good. And obviously sugar, as you all I'm sure know, is a natural antifreeze. So one of the benefits of actually eating stuff that was grown in Canada is it tastes better, especially in the winter months, because it tends to be sweeter. So we had some stems that looked great. They looked like they'd done well through this deep, well, -10, -15. For us, it was, sorry, Dan, but for us, it was cold. And when I picked them to taste them, I realized that there was actually some crystallization within the stem. So now that things have thawed out, you can see those stems, the core part is still okay, but those exterior stems just didn't have the sugar content. And they tasted good, but they should have been sweeter to survive the cold snap that we just had. So yeah, don't do it scientifically, but I'm always nibbling in the garden. Yeah.
Okay, great. Here's a here's a question for any one of you. And it's how do you trace the crossbreeding lineage, or how do you track the experiments in your field? This came from Andrew.
Well, I can answer that. I guess there's two different parts in there: what's happened in the past and what's happening currently. So in the field, we put out stakes for all our seed crops that have the product code, if we have one, the name, and then a lot number that we've assigned. And we assign the lot number when we're crop planting, decide what we're going to grow. Everything gets a unique lot number, and we'll follow that along with it. So that's how we track in the field. Now, for breeding lines, when we harvest something, we will give it a 5-digit code, and it'll have the 2 first digits will be the year that we harvested it in, and then the other 3 digits will let's say it's this. If we harvest a radish this year, it'd be like 24001 would be the first one, and we have a set of codes for the radishes, a set of codes for like a brassica rape, a set of codes for the lettuce. So we might have a number of 24001s. And then the next year if I'm growing a radish, it'll say radish 24001 that I've grown out. So I know what it's coming from. And I have a spreadsheet that says what I selected it out of. So I can go back to see what 24001 came out of. And if so, I have 24001 and I have it's a lot of diversity, and I have something I like that pops up out of it, I might have a 24001-1, 24001-2, 24001-3. And then, when I harvest them, I label them that way, and then put in my spreadsheet that way with whatever characteristics I want to record. And then I grow it out the next year and be like 25001 would be coming from 24001-1. That being said, I don't tend to go back often to see where things came from unless I've gone wrong. If I have a really big problem and I've lost stuff, either my fault or just the genetics, then I'll sometimes go back to see where it came from and maybe try to go back to a previous generation to get some remnant seed and start again. But usually I don't tend to go backwards too much.
Okay, great. I had a feeling a spreadsheet was part of your record keeping method. I just wanted to invite Chris. I know
I just wait on that thing that I was very inspired by John Navazio. So this is something that he kind of showed me along. It's not something I came up on my own.
Okay, great. I just want to invite Chris. Feel free to jump in. I know you're not able to have your camera on, so if
Yeah.
there's any question that you want to jump in on, feel free.
No worries. Yeah. I mean, I would say that I also use spreadsheets. My husband teases me for my spreadsheet use at this time of year, especially. But yeah, I tried out doing sort of complicated lot numbers and have kind of dialed it back a little bit because it was not something I ended up using. But maybe I could organize things, maybe if I was more of a spreadsheet expert. But yeah, we label things really well in the field, and I do keep track of the year. And if there's any sort of different lot numbers from each year, like if I'm keeping seed from the best plants, then that sort of gets a 1 next to it kind of thing. So keeping track of things is pretty important. When it comes to the pepper and the tomato that I shared, we kind of lucked out knowing like what the father was of each one, just because it was very clear and obvious. And so that was just pure luck. I mean, we've definitely seen crosses that I don't know exactly, like the red paste tomato that popped up in a variety called banana legs. Banana legs is a yellow determinate paste tomato, and it was clearly not a mix up. You know, every now and then we'll have an accidental, you know, where we accidentally mix something when we're packing or cleaning seeds and stuff. That doesn't happen too much, I'm glad to say, but every now and then. And yeah, so that one I don't really know. I have no idea where the father genetics came from. But yeah, so that's sort of how we can kind of tell what's happened in our field thus far. Yeah, I don't know. Unmute, or I'll mute myself again, but that's what I would say.
Okay, great. Yeah, just feel free to unmute yourself and talk up anytime you want.
David, I've got a seed crop record sheet. I can just share my screen for a second here. So, oops, I don't know if you guys can see that. So I did my plant breeding internship years ago with John Navazio. Was pretty lucky to do that. Thank you, Bauta, for giving me some funds to get a plane ticket. And yeah, I agree with Dan. John has excellent record keeping for all the crosses that he's doing. And so over, I guess, the last 15 years of more seriously growing seed and making my own crosses of things, this is the record sheet that I keep for the different seed crops. And it, you know, obviously I'm adding to it each year. Some of them I've been growing for 15 years now. So I do a varietal and subcategory. So the category might be tomato, the subcategory might be cherry tomato, and then this would be strawberry cherry, which is one of the new varieties that we've created. It's a wet seed crop, solanum, obviously. And then it's an annual. I put in the location where it is in the garden in case we get disease issues or something else cropping up. We know where things were growing. And there's a lot of information. You don't necessarily need that for all your crops, but if you do something like this for a crop when you start growing it, you've got that information to go back to. And then looking at whether it required roguing, if so, how much, why did you have to rogue? You know, maybe you realized, oh geez, when I grew it out 2 seasons ago, it was too close to something else, and this is maybe what happened. But as other people who are into seeds, like Dan will know, that's sometimes when you're roguing, that's actually where you get your most interesting stuff happening, and you see these opportunities of new varietals. So if someone's interested in this, I can send it to you, David, and you're welcome to pass that out. It was a huge amount of work to put this together. So if folks can just acknowledge where the form came from originally, that would be helpful. And yeah, so yeah, happy to share.
Great, thanks. Yeah, I'll jump on to the next question here. It's to do, again, for all of you, it has to do with making selections. Does anybody have experience on making selections based on nutritional content? And maybe a somewhat related question that came from the registration page is somebody asking about seed saving and contributions and selection from like a varietal acclimation perspective. So, you know, hardiness or appropriateness for your own zone, but maybe the nutrition question first.
We don't do any kind of nutritional analysis as such, but as I've understood, and I am not a nutritionist, is that often flavor is an indicator of nutritional content, and we look for flavor, but that's the extent of that. I Do you want me to talk about hardiness right away? Or you want me to let other people talk about nutrition first, David? You're muted.
I can talk about nutrition briefly because my other work when I'm not farming is I work as an ethnobotanist, and my specialty is revitalizing traditional plant food systems, both indigenous and non-indigenous. And the science is solidly that flavor and color are often related to nutritional content, whether it's the phytochemicals like the anthocyanins that are the pigments that are anti-inflammatory, anti-free radical, anti-cancer. And the richer the flavor, I don't confuse that just with sweetness, which is the only flavor we're hardwired for as a species. But yeah, if you get a butternut squash and you open it up and it's got a deep orange flesh, the pigments in there nutritionally are very important for you. And if it's got a rich, complex flavor, you do tend to have a greater range of nutrients within that, obviously depending on the species that you're working with. And yes, I select for color and I select for flavor.
I was going to suggest, yeah, let's stick to nutrition, and then if we want to go to the other one next. If nobody has any other thing to say about the nutrition, though, what do you do for climatic acclimation?
Get rid of stuff that doesn't survive. Yeah, just let things die and be okay with it.
I think that's really where it's at. We have a mix that we call our wintergreen mix, which is a bunch of brassica raypas, and essentially, I guess a good starting point sometimes for breeding projects for stuff that's a little bit more resilient is things that have surprisingly survived when they shouldn't have at all. And like in our first year of our farm, we had a cold frame, so like just a big window on some 2x4s that we were growing in early April. And we had a bunch of brassica rapa in there, and we harvested some, not much, and they went to seed and kind of ignored it. And then the next couple of years, it kept coming back in that patch, but only some of them. And that's something that I harvested from and then have since intentionally been selecting out. And so they're much hardier than the other brassica rapas. And so that's, yeah, just put them in tough situations.
Okay, great, thanks. It's hard to know which questions to go to next. There's a question here from Lorena, and I think I understand what you're asking, but maybe you can clarify if I get it wrong. Lorena's wondering if there's a plant that grows a lot smaller than it should be due to various factors. Is it still going to produce normal seed under normal conditions? Therefore, you're getting the same DNA. So I guess what I'm hearing is if you're growing a bunch of maybe you're growing out one of your breeding lines, and there's somewhere in the field where just none of the plants really got big, probably because maybe it didn't have enough fertility or for whatever reason. Is it still worth collecting the genetics from that? And Lorena, feel free to correct me if I misunderstood your question.
I don't mind jumping in here, just to share our experience, because we kind of have a tough love kind of approach at a lot of the time. Just partly with the conditions. We've had some real years of drought conditions, and like this past year was the opposite, swinging. And yeah, I find that from the perspective of, you have a tough year or maybe a tough few years, I find that our seed kind of gets that resilience. I grew, my example would be squash, which we've done a lot of varieties of squash over the years, and I do a lot of hand pollinating in a typical year. And we had a year of drought where just everything was smaller. The fruits were smaller. We weren't able to irrigate. We kind of do survival irrigation, just bare minimum. And so all the squash fruits were smaller, and I saved the seed, and I was wondering that very thing, like, am I selecting for it to become smaller fruiting? And then we just find out from people who then grow our seed and baby it and get treated the way it really wants to be treated. And they report back that, you know, and I even see I'm thinking specifically of a long pie pumpkin, which is my favorite pie pumpkin. And we have some local market growers who are really good at growing vegetables for market. And they were showing me the result of our seeds, and they were big, you know, and I knew that the seed had come from at least 2 generations successively of drought conditions where they were undersized for us. So, so I'm curious. I mean, I think that probably there could be some, you know, genetics that kind of lead to maybe dwarfing or something over time. I can't really speak to that because I'm not a geneticist, but what I've found is that yeah, you can select, like, we had some turnips this year that just were at the end of the row, that the soil was saturated, barely any oxygen, and they were little teeny but beautifully shaped turnips. And I knew that their genetics were good. They were just growing in a really crappy condition. They weren't like, you know, sometimes you get just tiny and really malformed root crops that you know that there's just bad genes in there. These were like perfectly formed little miniatures. So, so in my experience, I would say that in general, the DNA is good out of, you know, that sort of scenario where they have tough growing conditions and manage to produce. Maybe someone else has, yeah, more to add to that.
I actually have some experience just when I do selection in one of our fields, the whole bed is a bit sloped. So one end always grows bigger stuff than the other end. And when I do selections in that bed, I don't go to I don't just select from the big stuff. I select maybe the biggest stuff from the big stuff, but also the biggest stuff from the small area, because you can kind of tell it's more growing rather than genetic. So I don't know if other people do similarly, but yeah.
Yeah, what I've learned is that choosing the best stuff in your worst area is what's going to have the best genetics down the line. I think one thing that can, like, you know, if the seed gets fully mature, it's going to probably produce something good. But if you are always in the tough situation, always have things stunted, you might not be, you're masking the stuff that you're selecting for. So you're not actually selecting for, you're only selecting for what you're seeing there. So if there are potential good stuff and you do many generations in this in those situations, you might lose some of the good stuff that you'd like to see. But if it's in some years that you're seeing, you know, it's not visible, in other years it is visible, then you can keep working that way. But it is possible to, especially if you have a crosser with a lot of genetic diversity, it is possible to lose some of the traits you'd like to see.
Okay, great. There's a question here for you, Craig. Just because you were saying, like, now you're getting into researching some of the genetic stuff, and they're wondering what are some of the trusted sources for genetic research that you were talking about, and how much of this kind of reading do you do before you start playing? And I think what we heard is you didn't do a whole lot before, but now you're maybe
No,
so.
yeah, I didn't do any reading when I started this. It was just a seat of the pants kind of thing. But then, you know, you have questions, and so you go searching for answers, and that's kind of where it led me. But so like I have some textbooks for just like general genetic like foundational concepts and stuff. I really like Carol Deppy's book, the Breed Your Own Vegetable Varieties. And aside from that, I've been having a hard time finding stuff, but I mostly look for papers, like scientific papers and experiments. And you don't always find precisely what you're looking for, but often you can find at least some of what you're looking for. And then sometimes you'll find somebody that's running an experiment, which is exactly what you want, or like, oh, somebody sequenced the, you know, the genome for this crop, and you've got a list of all the genes and what they do. But yeah, it's been kind of tricky. I also would like some advice on how to get more info about that, but it's mostly I'm just like, yeah, searching online for studies and asking people that know more than me where they're getting info, and then just kind of, yeah, following that.
And yeah, correction on the question. It happened after Craig's presentation, but it is open for everybody. So you guys have resources you'd like to share, that'll be great.
Yeah, that book's awesome too.
And is this resources for breeding, or resources just for seed in general?
I'm gonna interpret it as genetics, like maybe resources for, which is kind of seed in general, but oh, breeding. There was the person's wondering about breeding.
So I know it's important, and I have studied genetics in university. I know that stuff. I kind of don't care, and I think I really respect people who put a lot of stuff into it. I'd rather just see what's happening and work with where it's going. That being said, Craig mentioned Carol Deppy's book, Breed Your Own Vegetables, which is phenomenal. It does have great stuff. Rowan White has a book also about breeding, plant breeding. You might be able to get it as a PDF now. I'm not sure. But I don't know if that's as much about the genetics, but those are two resources.
I like the way that John Navasio's book is divided, like with the amount of information that's in here. So for each category, so brassicas, for example, he divides it into each section, like corn or brassicas, whatever, has a general introduction to the crop. It talks about family characteristics, reproductive biology, life cycle, seed harvest, climatic adaptation, isolation distances, seed cleaning. So I've always found this a really good primer. And then as Dan says, just working with the seeds themselves is when you learn the most, and you pick it up pretty quickly. Like each crop is so different, and it lets you know quite soon. David's smiling and nodding his head too.
People, one thing that
I'll just say one more thing.
well, I was going to say, the more you talk about your work, the more people will send you scientific papers about the genes and will tell you things. And so I find that's a good thing, is to be public, and people will tell you some of the stuff that you find very interesting to know.
I was just going to say it's a lot easier to find information on like breeding in general than like specific genetic stuff. It's the genetics that I find more elusive. So yeah, you might have a harder time digging that stuff up.
Great.
Can I say one? There are quite a few plant breeders on Instagram that showcase the work, and if you send them questions, they will respond to you. And some of them know a lot of that stuff.
There's just somebody was wanting the book that you held up, Fiona, that was The Organic Seed Grower.
Yeah, John Navasio.
John Navajo. Yeah, somebody was wondering. Also, I just wanted to, we are kind of coming to the end of our time. I feel like we could ask questions for another hour. But just to respect everybody's time, I think we'll probably start to wrap up. There was somebody was wondering about access to your spreadsheet, Fiona. Would you like them to contact me and then I can reach out to you, or you want to share your contact in the chat, or?
Not that technology is not my forte. I work as a survival expert. If you're lost in the woods, I'm your gal. If you got computer issues, don't. With Zoom, does it allow you to upload files in the chat?
I don't think so. So, I mean, if people want to reach out to me, if that's easier, I'm happy to throw my email.
It says file. Okay, let me play with this. Let me see if I can figure this out. And if not, David, then I'll send it to you.
Okay. And so if people want it, feel free to reach out to me. I'm going to I'll put my contact email in the chat. And if you wanted that sheet from Fiona.
Okay. Yeah, maybe let me see if I can figure this out. Otherwise, I'll.
Okay.
If you put it in a Google Drive or in a Dropbox folder, you can also get a share link from that. if you have those things close at hand.
Yeah.
That's
And-
why so, okay.
I just want to let people know there's a link in the chat. It's a feedback form for this session, and we'd really appreciate if you can just click on that and let us know if this was useful for you, and then we know what kind of sort of training and education to bring forward. And I think what I feel like I hope people get out of this is that I really appreciate the experience that you guys all bring to this, and you don't need experience to do seed breeding. You just need to do it. And I think that's what I heard most of you saying. You can do it first and then get the experience while you do it. So I encourage everybody to just sort of jump in and yeah, try it on your own. In fact, there was one last question. There was a question on if people had experience in onion breeding, and there's probably not time to get into that, but on a related note, there was somebody in British Columbia who is moving farms and is not able to take their germplasm with them and is offering a red globe onion that they've been breeding. It's Wayne from Omega Blue Farms. If you're interested in some of that seed, again, my email is in the chat. Feel free to reach out, and Wayne is happy to share that seed. It's apparently an onion with a deep pungent aroma that stores so well that it's even hard to wake up in the spring sometimes. So that's the description. And if people are interested in accessing that, then feel free to reach out. And yeah, so I'll just say thank you very much to everybody. And yeah, it's great to have you all come and share your knowledge. And we will see you next time.